This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV. This Week in Virology, episode 1267, recorded on the 31st of October, 2025. Happy Halloween, everybody. I'm Vincent Racaniello. You're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
It's good to be here on this spooky Halloween, and here in Western Massachusetts, the storm clouds are thick and the wind is howling like a werewolf.
Where's your black cat, Alan?
He was around. I was hoping he would be on. We have two black cats. Well, the fluffy one and the short-haired one. And the short-haired one was, playing fetch and everything, and then he just went off on cat business. Anyway, it's 54 Fahrenheit, 12 C, and a cloudy, windy day.
All right. I am wearing the only mask that I have, a KN95, which actually has a copper lining. I'm going to take it off now because it's annoying. It's a reusable mask with a copper lining, which you can replace, and a little foam here, so it's very easy on the nose.
Nice.
Use that for airplanes. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. I don't have a whole lot of Halloween-specific stuff here. I will say that today marks, I may have said this in the last episode, today marks to the day 9 years since I arrived in Gainesville. Not Gainesville, Austin, Texas. So we arrived on Halloween in 2016. I will also tell you that it is a delightful, sunny 74 degrees, enough so that we've actually got a window open, and my cat, who's not black, he's a mackerel tabby, is sitting over in the windowsill enjoying the fresh air from outside.
We have two guests today from Ann Arbor, Michigan. Who are you?
Well, I'm Marge Simpson. I am here visiting Kathy in Ann Arbor, and I have to tell you, the weather is very much like Springfield. Today, it's 52 Fahrenheit, and that's 11 Celsius. Kathy tells me I have to tell the temperature in both Celsius and Fahrenheit.
Oh!
And, hey, Homer's not really here. Anyway, the sky is just like in Springfield. It's blue with white puffy clouds. So it's really fun to be here with you all. And I have to tell you that when you start talking about virology, I'm going to go away and let Kathy come in because I don't know as much about virology as she does.
Okay, well, thank you for joining us. It's a pleasure to see you. We have another guest from Madison, New Jersey. And who might you be?
Hi, I'm Ray. I'm taking a quick break from being on Tatooine. It's a lot colder here than it is on Tatooine. It's only 52 Fahrenheit here. But I'm still here and I'm ready to hear more about science.
Do you know anything about viruses?
I only know a little bit about viruses, but I bet I could stop them with my lightsaber.
I guess there are viruses on Tatooine, right?
There probably are. Very sandy.
Yeah.
Oh, sandy, arena viruses.
Huh?
There you go, folks. Happy Halloween. And Rich and Alan, are you going to take off your masks now so we can see who you are?
No, mine's stuck on a little too.
Yeah, really
too firmly.
funny.
If you like these programs where we lightheartedly do things from time to time, we talk mostly about viruses, and on the other programs, other things as well. But the key is we give you science, just the facts, nothing else. We discuss papers and so forth. And if you like that, which you need today, because you just don't know what's right. You have people spouting off non-facts all the time. So we'll tell you the facts. And if you want that, we'd love your support to continue to do it. Go to microbe.tv/contribute. Don't forget Immune 100 live at the incubator, December. I don't remember. Do you remember the date, Brianne?
22nd, maybe?
December 22nd, 6 p.m. You have to get tickets because there are only a certain number of seats, and we have sold a bunch. So if you want to come, you better get moving because I think they're all going to sell out in November. But go to microbe.tv. At the very top of the page, there's an announcement for the episode 100, and you can buy tickets. The tickets are a donation so that you could tax deduct them, because MicrobeTV is a nonprofit entity. So we will have the entire crew of Immune, including Ray with her lightsaber.
I can bring the lightsaber.
I have one at home, too. I could bring. With my black shirt, I could be some
Oh,
character.
you definitely could.
Well, with a mask, almost Darth Vader, right?
Yeah. You just have to breathe really loudly. Perfect.
Not bad. Not bad.
Not roomy enough. RHEUMY. Right?
Yeah.
Anyway, and also all the Immune crew will be there, and we'll have 2 local guests and some surprises here. So if you want to celebrate with us, please come. And I guess Alan put these stories in, right, Alan?
Yeah, I found a couple of news stories that are kind of relevant. So the first one is linking to a story in Science that long-tailed macaques, Macaca fascicularis, we've talked about them before. These are a common, as common as primate research is in virology, these are used as a non-human primate subject for a fair number of experiments. They were classified, their classification internationally for endangerment went from, I think, threatened to or vulnerable to endangered, officially endangered a couple of years ago. And a group of biomedical research folks lobbied for an appeal to that, argued that they shouldn't be classified as endangered, but the IUCN has held up the ruling, and these monkeys are officially endangered, which is probably going to complicate research on them.
It's a great picture in this article.
Yeah.
Boy, those eyes, they're so intense.
Yeah.
Wonder what they're thinking.
They're
Did you
thinking
see-
they're endangered.
They're thinking they're
Yeah.
endangered.
Yeah. Yeah.
This one is in Cambodia. You heard this week that a truck full of rhesus macaques overturned in Mississippi.
I couldn't remember where, but I heard somewhere that
So they were raised at the Tulane Primate Research Facility. So there are a number of NIH-funded primate centers throughout the U.S. There used to be one in New England. It was shut down. And they provide primates for other institutions for research. And this one was driving 20-some-odd rhesus macaque somewhere. The truck overturned, and
Wait a second. I saw this movie with Bruce Willis and
yes, right. What was that
Brad
called?
Pitt.
12
monkeys.
12 monkeys, right. Well,
Great
this was
movie.
20-some-odd. And most of them were killed. I don't know if they were killed in the truck accident or captured, but 3 of them are still running around. They shouldn't kill them. They should put them in a sanctuary. They should capture them and put them in a sanctuary.
They
isn't
should.
it?
Yeah.
But, it's just unfortunate that we don't value animal lives as much as our own.
Well, no,
So-
it's normal that we don't value them as much as our own, but we should be careful with them anyway.
So
Well,
what I don't understand about this, the macaques that are used for research,
hmm
are they not raised?
Some of them are raised, and so that's one thing that the article doesn't really get at is what's going on in some of the places that are raising those primates.
Yeah.
But they do sometimes import groups of primates into those places. Usually there's some spots in the Caribbean where they will raise the monkeys and then move them out into some of the other primate centers.
Also,
Okay.
I think there are some restrictions generally agreed upon for breeding endangered species.
Yeah, there's
That
not a
may-
ton of breeding in most colonies, at least the
Right.
ones that I'm aware of.
Right, but if they're being raised in captivity instead of captured in the wild, you would
Well, the ones in the primate centers are all bred.
yeah.
Like the one in Tulane is many, many thousands of animals.
Some of them are imported too.
Yeah,
I have
different
to say that
species, I think.
speaking for myself personally, this is strictly personally, I'm really conflicted about all this stuff.
Yeah.
I have a hard time. I have a hard time with the whole thing. I don't like inflicting pain on the animals. I understand the justification. I wish there was a comfortable middle way, but it's hard. It's hard. the whole thing is hard.
Well, and I think researchers in general are not big, like, fans of working on animals. You don't hear people say, "Oh, good, I get to go, inject the monkeys," or "I get to go," even mice. It's like, it's not, and it's not just that you're hurting animals. It's, there are limits on the science you can do with it. It's just the least bad option that we have. And we say many times on here that, oh, well, this happens in mice. We don't know if that happens in humans. We'd like a better system. And some of these things like organoids and such are kind of moving in that direction, but we're not there yet.
Yeah, I mean, there are some experiments that can only be done in these systems.
Yeah.
And some of those I think are interesting and important experiments. And I certainly wish there was a better way of doing it. And so we have to kind of think carefully about, doing things as ethically and as, only as necessary.
Yes.
And Vincent, you've served on animal use committees, right?
Mm-hmm.
This is a justifying animal use is a big deal, right? You have to basically say there's no other way to do this.
Yes, absolutely. Even with the smallest, with mice, you still have to justify not only the use of the species, but the numbers that you're going to use, right? You can't just say, I need 5,000 mice for this. Well, tell us why you need it. And it's a lot of work, and they scrutinize it very carefully.
And that's,
you can imagine larger animals even more so.
Yeah, the things
Yeah.
that had to happen in working with primates when I was a graduate student were pretty intense.
And that the quantity of animals is something people are also working on, because a lot of the newer sensors that we have and newer data analysis techniques, you can get by with using fewer, which is great for everybody because it costs less and you get, better results potentially with fewer animals. And so there is progress, but it's not
Just
done yet.
remember, mice lie, monkeys exaggerate, and ferrets are not humans.
Yes.
Right.
Oh,
So
here's
I
my
just-
Halloween cat. Theo,
I'm
Theo,
just
say
not
hi to
convinced.
everybody.
Hello, Halloween cat. I'm not convinced that primate studies are all that useful. I know that for some viruses, that's the only model, right? But make a smaller model. And the mice are not also parallel either, but I don't know. I have problems with the primates and
Would
the
you-
cats and the goats and the dogs.
Would you accept a drug or vaccine that's never been tested in non-human primates?
Yeah, I would.
Okay.
I would do that. Sure, I would do that. Any other animal would, a mouse would be fine. Yeah.
Okay.
But you get results in mice and they don't. It would
They
be
don't.
lovely if we could do a mouse experiment and not have to do a clinical trial, right? But we can't yet.
It would be even better if we could do an organoid experiment or a cell on a chip experiment, not have to use any of this and get results with human cells.
NIH wants us to use AI to replace animals, which it's
Okay.
going to take many years.
It's going to
do
take
that.
years, but you can combine that maybe with some of these other cell-based systems and reduce and keep reducing until we get to the point we don't need them.
I mean, it's certainly worth investigating, but
Yes.
it's not ready yet.
No.
For sure.
Yeah, that's my worry is if we really know enough yet to actually make that switch now, which
Right.
I don't think we do.
So there are mosquitoes in Iceland, Alan?
Yeah. So the other thing, this just popped up in my news feeds. Iceland has reported for the first time that they have breeding populations of mosquitoes.
Wow.
These are Culiceta annulata, so it's not one of the usual species that we talk about, but these are mosquitoes, and they've had, of course, mosquitoes get brought into Iceland periodically on cargo and such, but they don't survive because it's Iceland and it's cold and they freeze to death. But these apparently are surviving, and
So I saw that it was something like 2 males and 1 female. Is that what constitutes breeding population?
think right. So I think they what was it? Yeah, I think it's mainly that they don't have any other obvious source for these.
Yeah, it was 2 female and 1 male. That's right.
Yeah. And this particular species is not known to carry anything. But as the climate warms, Iceland and other places in the north are going to get affected.
So Kathy, this is endangered, the species in Iceland.
Oh,
Right. The species is endangered in Iceland.
Because now the last 3 are gone. Actually, they're extinct in Iceland.
Right.
So this guy says, on the evening of October 16th, I caught sight of a strange fly on a red wine ribbon. It turned out to be a mosquito.
Yep.
Yeah, there may be more. Do you think it's related to the climate warming a bit, or doesn't have to be warming?
That's what the article says.
Yeah, I mean that's a reasonable theory. It could you could argue, well, maybe this is an unusually warm year or coincidence or something, but this is the kind of thing that we're sort of expecting to happen.
Yeah, it says that these mosquitoes can live in cold weather.
Yeah.
Normally, so maybe it was just a matter of them getting to Iceland.
Yeah.
Okay. Well, if there's mosquitoes, I'm not going to Iceland.
Right, because there are no mosquitoes in New Jersey.
Wow, I had no choice of living in New Jersey, although I could have moved many times, I guess. But you know what's very interesting, Alan? Mosquitoes don't bite me.
Really?
Because they're too busy biting me.
They do not bite me. I can be amongst people who are getting bitten through their clothes even, and I am unscathed. So I am repellent for mosquitoes.
Are you sure you're unscathed, or are you just not developing the welts?
Well, I don't have any welts. That's true. Or, maybe one per summer.
Because not everybody has the same reaction.
Yeah, so they
And
can
in
bite
fact,
you
people
and
who've
you
been
don't get a
bitten a
welt.
lot will sometimes get tolerized to it.
Oh, okay. So
Yeah,
it's like an asymptomatic infection.
It's
I
like,
never had
yes,
COVID.
You
right?
may
Yeah,
be getting
it's an
asymptomatic
allergy,
mosquito
yeah.
bites.
Okay. Well, some people could see them landing on them. And
Oh yeah,
smacking them,
I
so
see them
I've
landing on me
never seen
occasionally.
But they are attracted to certain people and not others.
Yes.
Yes.
For what gradients of carbon.
Apparently my
dioxide.
grandmother was extremely attractive to mosquitoes, and my grandfather, who was an entomologist,
Was not.
sometimes enlisted her help in catching them.
That's cool. All right. Good. Those are lighthearted. Well, not so lighthearted.
Yeah.
We don't want to endanger macaques, but at least they're not politically charged news items. How about that?
Yeah, that's good.
Although we could,
Oh
because
yeah.
it still continues,
no, let's
folks.
not yet.
We
Yeah.
will.
No.
We will do it another time. I'm sure the letters will get to it if we get the letters. Okay, we have a snippet in a paper for you, and so the snippet I picked is a meta-analysis, which is not where the authors did laboratory research, but they did collect other papers with laboratory research, and I thought this was an important enough topic that we should do it. So, Kathy, do you want to give us a summary of that?
I
Sure.
can do authors if you like.
Oh, sure.
Yes, please,
Sure.
Alan. Sorry.
Yeah, so the paper is open access. It's published in Viruses, and its impact of vaccinating adult women who are HPV positive or with confirmed cervical SIL, that's squamous intraepithelial lesions, with the 9-valent vaccine, a systematic review. First author is Dominik Pruski. Senior author is Marcin Przybilski. They are from the District Public Hospital in Poznan, Poland, Jagiellonian University Medical College in, is it Krakow?
Krakow. Okay.
Krakow,
Yeah.
Poland, and Poznan University of Medical Sciences. So it's an all Polish team.
So, as Vincent said, this is a meta-analysis. So what they do is they look at a whole bunch of studies and figure out what can be gleaned from the data by sort of looking at all of them en masse. And what they wanted to do is see whether there's a positive benefit of vaccinating people who are already positive for human papillomavirus, also known as HPV, which causes cervical cancer. And so the vaccination would be with the HPV vaccine. And so instead of using it as a prophylactic vaccine, using it as one that's therapeutic because the person is already HPV positive. So as indicated, these investigators are in Poland, and it turns out that in a number of countries, the HPV vaccine has been given to people who are HPV positive or have confirmed precancerous cervical lesions. So we'll talk a little more about what those precancerous lesions are and the various grades of them. But if someone is HPV positive or has these lesions suggesting they might progress to cancer, then in some countries, even if it was way past the age of when they could have been vaccinated, they can give them HPV. And so the authors did an extensive literature search. They started with 653 possible studies, and they narrowed that down to 7 that met their criteria. And they even graded the studies based on the quality of the experiments and the data and analysis and so forth. And these all got good scores. And the result was that vaccination was linked to reductions in persistent HPV infection, linked to reduced progression of precancerous lesions, and reduced recurrence of high-grade lesions after surgical removal. And so their conclusion is this warrants further study, but the results suggest that the earlier the vaccine is given in such HPV-positive people, the better. So they compared studies where the vaccine was given before a treatment at the same time as a treatment, which is usually some kind of a sampling, a surgical removal of some of the tissue, or even after surgical treatment. And the sooner they gave it, the better. And so I have to say that for me, this was kind of news that you could give this vaccine in this way. And I checked with a friend and neighbor who's an OBGYN and said, does this really happen here? And they said that yes, such treatments are used in the United States, although it can be tricky sometimes for getting insurance coverage. So they have their patients check for that coverage first. But basically, their knowledge of it without having read this paper was that it seems to be have some positive benefit in reducing some future development. So there were some hints of that out there. And the 7 studies that they have, 2 are from Spain, 2 are from Poland, 1 from Denmark, 1 from Czech Republic, and I can't remember the 7th one. But anyway, the thing is, in the United States, the CDC doesn't have any data recommending this, the vaccine for adults, but they do allow shared decision-making, which you've heard Daniel talk about on the clinical update quite a bit.
One thing about the U.S. recommendations for this vaccine is that when it was originally approved by the FDA, it was approved for women ages 9 to 26. And the recommendation has actually broadened quite a bit. And so if you, so of course it is now recommended for men as well. And if you were a person who was not in that 9 to 26-year-old age group when it was first approved, because of the broadening, you may now be eligible. And so that actually is what happened with me. I wasn't eligible when it was first approved, and then they broadened the ages, and I was. If you are, so it's great that this is working therapeutically, and if it is also possible that it may be a prophylactic for you, it is likely might have been increased eligibility for you.
Right. So one of the other reasons I did this, because besides that I picked this paper, besides that it's an important thing, is that a couple of weeks ago someone said to me from a European country, I just had a test and I'm positive for all these high risk HPVs, and I'm going to go get a HPV vaccine, and the doctor told me it would clear the infection. I said, what? I've never heard that. And so I Googled it, and Google, in all its wisdom, said, no, HPV vaccination does not clear infection. So I didn't say anything to that person. Okay, you should get vaccinated anyway, because there may be different serotypes and genotypes. And then this week, this paper pops up, which suggests that, yes, you can, and as you'll see the numbers, in a certain percentage of individuals, the HPV is gone after vaccination. So,
And
Can I do HPV real quick?
yes, please do, please.
Right. Double-stranded DNA containing virus, circular genome, pretty small, very small, 8 kilobases, a naked protein capsid, There are hundreds of different types of HPV in humans, and there are HPVs in all other vertebrate creatures around that I'm aware of. In humans, they are primarily responsible for warts. The reason that there is concern, or what they can be really notable for, is causing, beyond warts, I would say, cancers. And this includes, most notably, cervical carcinoma, and it seems to be probably the sole cause of cervical carcinoma, and is also responsible for a number of head and neck cancers. In the case of cervical carcinoma and some of the head and neck cancers, the transmission is direct contact, mechanical, and in the case of some head and neck cancers and cervical carcinoma, it's sexual transmission. Some of the types are nastier with respect to their propensity to cause cancer than others. The mechanism of causing cancer is important. These viruses infect epithelial cells because they don't have a lot of their own replication machinery. They stimulate the cellular replication machinery, which causes, among other things, the cells to proliferate, and that's ultimately what causes a wart, hyperproliferation of the cells that are producing the virus. Occasionally, and this is not part of the replication cycle, the genes that are responsible for triggering that proliferation accidentally integrate into the genome. Now you're not doing virology anymore. You're doing cancer biology, because now you have cells that have these replication and proliferation stimulating genes inserted into them. There is a vaccine that it's been developed over time, starting with the most dangerous papillomaviruses, and now including up to 9 or even more. This study uses Gardasil 9, which covers all of the oncogenic papillomaviruses and a couple of others that are responsible for some head and neck cancers. Importantly, the vaccine is as safe as you can possibly get, right? It is a protein, period, made in yeast. There's no virus anywhere. But importantly, assembled into virus-like particles, okay? So it's as if you have the virus shell, looks to your immune system like a shell, with no virus DNA in it or anything. And it is a phenomenally effective vaccine. Correct me if I'm wrong, Brianne, but it blew the minds of immunologists in inducing an extraordinarily long-lasting antibody response, okay?
Yeah. And in terms of its sort of efficacy percent as well.
Yeah. And
Prevent
so,
cancer. Yeah.
and so it, over time, now we know that it not only prevents infection and the cervical, they're kind of like the cervical equivalent of warts. It's a proliferation of part of the epithelium in the cervix that will give terms like SIL and CIN as we go on through this. So they'll not only prevent that sort of less noxious disease, but as a consequence, since they will prevent the infection, they'll prevent cancer. So these are cancer vaccines. And the thinking up to now is that the best thing to do is to administer these things to individuals before they are sexually active. In the U.S., that's ages 12 and 13. Since it's a sexually transmitted disease that can actually cause problems in males as well as females, you vaccinate both males and females. And as we've already said, and this paper was news to me too. It just blew my mind. It turns out that they may be therapeutic as well.
Yeah, this paper, I have been thinking for years, it would be a good idea for somebody to do the experiment of vaccinating people who already have HPV and seeing if this does anything. And I'm so glad that this has actually been done several times and that the results turn out to be pretty darn good.
I mean, this basically means at any age you should be able to
Yeah.
get
Yeah,
It's
an
absolutely.
HPV
better
vaccine.
late than never.
Yeah, I think
And-
this was all news to me, and I thought it was awesome.
So famously, Henrietta Lacks had cervical cancer, and of course, HeLa cells were made from her tumor. And many years later, she had died a long time ago, but not too many years ago, the genome of HeLa cells the sequence was determined, and there was integrated into one of her chromosomes the E6 and E7 genes of HPV-18. And as Rich said, those are the genes that the virus would normally express to drive the cells to divide so that the DNA replication machinery would be available for the virus. But they accidentally integrated into the cell. The cells kept dividing, and eventually you get a cancer. Okay, so these HPVs are leading cause of cervical cancer, and as Rich said, anogenital and oral cancers as well, completely preventable, right, with the vaccine. And WHO actually has declared a strategy to eliminate cervical cancer by 2030, not far off, by vaccination, screening, and treatment. I actually did a TWiV in Stockholm a couple of years ago with a guy who works on HPVs and elimination of HPV and cervical cancer in Sweden. Very interesting. Put a link to that in the show note. So this WHO strategy, they want to get 90% vaccination coverage for women under 15, testing of 70% of women 35 to 45, and then 90% of women who have cervical cancer diagnosed. Well, that's the goal. And it's interesting. In Sweden, he said, I forgot his name, sorry. During COVID, they couldn't do pap smears, right? They couldn't have people go to offices. So they sent people swabs, and they did PCR for HPV. And he said, that's all we needed to know for cervical cancer anyway. Right? So HPV vaccination, of course, was introduced not too long ago, but still we have a lot of precancerous lesions and cervical cancers. Rich, did you paste that diagram of the different kinds of lesions? You want to give us an overview of what happens from infection to cancer?
Sure. I'll refer to that diagram. So the normal epithelium has a layer of cells on the bottom that are essentially the stem cells for the epithelium. They're an actively dividing layer. And then as cells originate from that basal level and migrate up through the epithelium and differentiate as they do so, to the point where at the top you have basically bags of keratin that are essentially dead cells that provide a physical barrier to insults from the environment. When HPV infects the epithelium, it does so through a break in the skin, and the virus will infect those cells that are essentially the stem cells in the bottom. And during that period of time, when they do so, they cause those cells to proliferate, as we've already discussed. But they don't actually make virus in those basal cells. As those cells that are infected migrate up through the epithelium, this is fascinating. They are subject to all the differentiation factors that normal epithelium has. And those differentiation factors, the virus is adapted to this, will trigger expression of the later genes that are responsible for making capsid proteins and that kind of stuff, so that in the upper layers of the epithelium, you're now making a load of virus that then is shed off into the environment. That's the normal situation. And if you have in the cervix, if you look at a biopsy of the epithelium from an individual who you expect may be infected, you can see an abnormal pathology in that you can see that in the lower layers of the epithelium, there's sort of more of these replicating cells. And there are terminologies for this. Either squamous intraepithelial lesion, SIL, and there's low grade and high grade, so LSIL and HSIL, Or you can refer to cervical intraepithelial neoplasia, grade 1, 2, and 3, 1 being the least problematic, 3 being the most. And basically, there may be other markers as well, but from a point of view of a pathologist, what you're looking at is how much of the epithelium is involved. Okay, if it's less than 50%, you're looking at low-grade SIL or SIN1. It's a mild or moderate dysplasia, or mild or very mild dysplasia. As more of these lower levels of the epithelium look abnormal, you increase to CIN1 or CIN3 or to high-grade SIL, from moderate to severe dysplasia. And during that period, once you get to high-grade SIL or SIN2, you're probably, you start to suspect as a pathologist that you're not looking at a normal virus infection anymore, that you're probably looking at this transformation that we talked about, which is the virus integrating and causing the cells to proliferate. Ordinarily, the virus infection is confined to the lower grade lesions. And ultimately, you'll get the entire epithelium involved, and in the end, cells will leave the epithelium, and you get an invasive carcinoma out the other end. As we've already said, the treatment typically is surgical. And there's a procedure called LEEP, L-E-E-P, loop electro-excision procedure, where they use a hot wire loop to excise the affected tissue, and like in most cancer surgeries, look at the margins and hope that they got everything. And typically, if you're low-grade SIL or SIN1 or something like that, they may just watch it for a while, because a lot of these, like a wart, will resolve. But once you get to the higher grade lesions, and now it also, they can type them and figure out, do you have just a not very nasty HPV or a nasty one? Okay, and that's part of the decision-making process as well. And once you get to moderate or severe dysplasia, or you have a high oncogenic potential virus, then you talk about surgeries. All of this is avoidable. Pap smears, vaccinations.
So that's where we come in here, that if you have had surgery to remove a lesion, sometimes a doctor will recommend vaccinations. They won't be covered by insurance typically, but there's no clear definition of when you should get it or who should get it. And it varies from country to country. So the vaccine that we're going to talk about in this paper is Gardasil 9. It has 7 oncogenic HPV types and 2 low oncogenic types that cause warts. And the access to this vaccine is not consistent all around the world. A few European countries offer free vaccination with Gardasil 9 for adults. Sweden gives vaccination to all interested university students. If I were a university student, I'd get it because it could save your life.
Yeah, if you didn't have it as a kid, get it as soon as you can.
Know, there's no downside.
No downside.
No.
That's
Zero.
so important. No downside.
Germany provides vaccination to men who have sex with men and immunosuppressed individuals. Other countries allow vaccination for adults, but it's typically not reimbursed, and it's not uniform across the globe. And as we said, CDC recommends HPV vaccine for children before sexual initiation, but there are no data recommending vaccination for adults. But for adults 27 to 45, the CDC and ACIP, the former ACIP, or the committee formerly known as ACIP, do not routinely recommend vaccination, but instead shared clinical decision-making, right?
And this is why it's not covered by insurance, because there's no consistent recommendation for it.
Right.
There was a line in this paper saying that the vaccine in some country, or maybe many countries, that noted that the vaccine uptake was much higher in situations where you didn't have to pay for it.
Yes.
Of course.
And that's telling.
It's a simple public health measure, right? You encourage vaccination by paying for it, and it's not that much money.
Right.
It's less than adding a wing to the White House, okay?
And it's also less than treating a person for a cervical
a lot
carcinoma.
less than surgery.
Yes.
Yeah.
Yeah, in fact, that was part of, what happened with my getting it, is that I suddenly found out it was something that I was in the age group and my insurance covered it for people who had never had it. And I was like, hi, sign me up.
Yeah.
I have everything I've read about this.
I bet you would have got it even if they didn't cover it.
I would have, but it came across my
Yeah, of
radar
course.
that, in fact, hey, this is a vaccine you haven't had that you're eligible for and that we cover. And I was like, oh, I didn't know I was eligible. Sign me up.
Yeah, I mean, it's $150 a dose times 3, right? Something like that.
So that's significant for a lot of people.
Significant for a lot of people. Yeah.
I understand. But if you
It's
have
significant
a choice,
for everybody, but it's a big deal for a lot of people.
Yeah, I understand. Asian countries, it's not systematic. They in a review on Asia Pacific region, many national immunization programs focus primarily on adolescent girls, and few offer adult or catch-up vaccination policy. So that's why they did this study, to do a literature review, and they look in databases and the first objective is to assess the impact of Gardasil 9 in adult women who are HPV positive or have confirmed SIL. And right now, there's no algorithm for HPV vaccination for HPV positive women, which stage lesion you would get it at, and so forth. And the secondary objective is to evaluate the role of vaccination in reducing the recurrence of a lesion after it's been excised, and whether vaccination influences that outcome. Okay. So in other words, there are research studies that have addressed these issues, and they're going to look for them and put them all together. So they use 3 databases of scientific literatures. They do searches with terms like HPV vaccination, HPV infection, cervical intraepithelial lesion, surgical management, and so forth. And then they have a table with all the search terms that they used. They got 25 studies. They looked at them. The authors sat down and discussed the inclusion and the exclusion criteria. For example, inclusion, studies on the impact of HPV positive subjects with the 9-valent vaccine, vaccination of adult women. Exclusion criteria, healthy population, unfinished studies, etc. Vaccination in children and men. This is only focused on women here. And then they have a way of extracting the data from these papers. They also do a risk bias assessment, as Kathy mentioned. The Newcastle-Ottawa Quality Assessment Scale to evaluate quality and potential risk of bias of studies.
And one very important exclusion criteria, they excluded other meta-analyses.
Right.
We're not going to have any infinite recursion here.
Right.
Inception. No inception.
No inception, right.
And so they identify 653 articles, and they end up with 7, and they make sure they're all methodologically good. And these 7 evaluate the impact of the Gardasil 9 in patients with HPV infection or precancerous cervical lesions. 19,414 individuals, 33% were vaccinated, and 66% were unvaccinated. And they have a table with all the papers here. You can see the paper and the aim of the study design. Some of these were retrospective, where they looked back at what has already happened, and some were prospective, where they're looking at the time of after vaccination to see what is happening. They look at the outcomes, the goals of each one.
They are all specifically a type of study called cohort studies.
Right. So they said that the papers evaluate the effect of vaccination differently. The Polish authors, of which they are them, right, or some, they're in Poland, were the only ones to determine the scale of HPV disappearance according to genotype assessed at the time of diagnosis and 6 to 8 months after the last dose of the vaccine. So whether the same genotype is present, whether you have partial remission or complete remission, a new genotype appearing or disappearing and reappearing. So very comprehensive study, right, looking at genotypes. Another study looked at tests at 6 and 15 months, but no genotyping there. And another study, the most extensive follow-up, they did early, medium, and long-term effects of vaccination follow-ups at 6, 18, 3 years, 6 months, 18 months, 3 years, and 6 years after cervical excision. up to 15 years. Wow. In some studies, they didn't look at HPV infection, but they looked at the lesion itself and so on and so forth. So they say the results of all the studies were similar. They indicate the benefit of vaccinating adult women after sexual initiation with HPV infection or already with cervical disease. For example, an Italian study showed that among women treated for LSIL, low-grade SIL, a positive HPV test result was obtained in 38% of unvaccinated women, compared with 18% in vaccinated women. And they say similarly, a spectacular effect observed in a group of women treated for high-grade SIL. 18% of vaccinated women had a positive HPV, compared to 8% in the vaccinated. So in the unvaccinated women, sometimes excision of the lesion will get rid of infection. But you can imagine that, the virus is going to have spread in some cases.
And HPV in particular goes everywhere. It just, yeah.
So then they had a study where they look at infection itself. So in Poland, researchers showed the rate of HPV disappearance was higher in the vaccinated group over the follow-up period compared to the control, especially in the case of complete remission, that is, disappearance of HPV. It was observed, complete disappearance of HPV was observed in 72% of vaccinated women compared to 45% of unvaccinated. And it was particularly evident when they look at the genotypes covered by the vaccine, right? Because those are the ones more likely to disappear. Also reducing risk progression. One study showed the incidence of repeat conization. So conization is the biopsy procedure. So if you were having it done again, that means there's some signs that something is going on. The repeat conization was 18 per 100,000 person days in the unvaccinated cohort, 2 per 100,000 person days in the vaccinated cohort. And that was found in another study as well. And as Alan said, vaccination compliance increased when the vaccine was publicly funded from 35.9% to 79.1%.
Yep.
It's really big.
Yeah.
Yeah.
It's really big.
Price and accessibility. It's just
Another study, SIN2+ recurrence was observed in 513 unvaccinated women, 14 prophylactically vaccinated women, and 15 in women vaccinated post-excision. So the recurrence within 6 months, again, of conization was reduced by 80% with prophylactic vaccination, 89% with incomplete post-excision vaccination. And among 1,700 women with a positive cone margin, the recurrence was identified in 272 out of 1,568 unvaccinated women. In the vaccine group, it was in 84 prophylactically vaccinated and 119, with only 6 recurrences in each group. Vaccination before 15 years had a 35% lower risk for progression to SIN3 or worse. Vaccination between 15 and 20 years had a 14% lower risk. So that's just a little bit of a summary of the data. It's really compelling.
It's amazing.
Yeah.
That, and we are only, Gardasil was only adopted in 2017, right? So it's still early days of coverage. And in Italy, catch-up vaccination is recommended for women up to 26 and men up to 18. They also recommend vaccination for women treated with SIN+, 2+ or higher grade, and for individuals with HIV.
Was it 2017 or 2007?
2017. Gardasil 9, that was the year that
the
that
9-valent.
Yeah, the 9-valent. In Poland and the Czech Republic, as of 2025, this July, adult vaccination is not publicly reimbursed, and patients wishing to get it must cover the full cost. It can be given to people up to 45 in many countries. Again, in some countries it's fully covered, and in some countries it's not.
Brianne, could you define cohort study for me?
Sure. I was just writing a homework assignment about that yesterday. A cohort study is a study where you take a group of individuals who don't have a particular disease and you put them in groups based on their exposure. And then you look to see whether so in this case, the exposure would be the vaccine. And then you look to see whether there's a difference in outcome. You can do this on old data on exposures. So that would be a retrospective study where you basically go look at past exposures and then look at outcomes, or you can actually do this at the beginning, start the patients with exposure or non-exposure and then follow them for 15 years or so, like this the prospective study that we have here mentioned.
So I would hope that this paper would at very least precipitate a whole bunch more such studies. I mean, I don't know kind of what the guidelines are for clinical treatment and whether, whether you need to initiate a trial. Probably depends on where you are. But if you have the option of vaccinating people and you have you have knowledge of this and you keep track of what's happened, hopefully there'll be a lot more of this to reinforce some of these data and ultimately change the recommendations. This is amazing.
Cohort studies are pretty high in terms of the rankings of how strong their evidence is for the association. Not as strong as doing something experimental, but sort of outside of that, they're about as strong as you can get. So this is a great data.
So,
and I think what I'd like to see is, groups, medical groups, like I guess it would be the American College of Obstetrics and Gynecology, ACOG.
Probably.
Is that I think that's a thing.
That's a thing.
They would be the ones in this country to take the lead, and there are similar societies all around the world who I don't know what their current guidelines are, but whatever they are, they should take these data into consideration and maybe make an official recommendation if you have patients who have these early stage or any of these stages, recommend the vaccine. And I think that's part of what kind of gets the ball rolling on reimbursement too.
Sure, absolutely. If these societies recommend it, it will help a lot.
Yeah.
So
ACIP is not going to recommend it anymore.
No. So as I was reading this, what I found myself thinking about was rabies.
Yes.
Because we often tout the rabies vaccine as the only vaccine around that you can give after infection and prevent disease. This looks to me like number 2.
I think we have another one.
Because we have essentially a persistent or long-term viral infection that you can come in with a vaccine after it's already been initiated and compromise the future of that infection. It's amazing. And also now, I think I've got this right, but Brianne, I'm looking at you.
Okay.
My understanding is that the normal immune response to a papillomavirus infection is primarily cellular, and there's very little antibody response. And yet the vaccine here it's mechanistically, it's primarily an antibody response, a very robust antibody response. So my sense is that we're fooling Mother Nature. Okay?
Right. I think that I agree with you. I think that the thing about the vaccination is that because it's in often cases trying to be prophylactic, having the antibody response to actually block the initial infection is more important. Whereas when you have established infection, being able to kill the infected cell with T cell response would be particularly important. I will admit I don't know enough about the VLP here to know if there are T cell epitopes in the particle or not. I'm ashamed to say that if you would ask me in 2019 about certain types of protein-based vaccines, I would have said, "Oh no, that's an antibody thing." And I remember being very surprised thinking about how many T cell epitopes were in spike. So I don't know if maybe there are T cell epitopes here as well. I don't know, but it is usually thought of as an antibody-inducing vaccine.
Data must be out there.
Probably.
And the structure as a virus-like particle, my understanding, is really important to have a high density of antibody epitopes on a particle, and probably a lot of them are structural epitopes as well. Having it look like the virus is really important.
So the authors conclude, they propose several practical recommendations. Adult women who are HPV positive or have histologically confirmed SIL should be offered 9-valent HPV vaccine, particularly when undergoing excisional procedures. The most significant benefit appears when the first dose is administered before surgical treatment. And that's easy enough to do, right? You're going to go in for surgery, you get the vaccine beforehand. But how long is a good question, right? And they say to strengthen the evidence-based, prospective, randomized, multi-center trials with standardized outcome definitions are needed to help confirm the observed benefits and guide consistent global recommendations. So yeah, I think they're right. You probably do need some more clinical trials, some clinical trials to be done to solidify that, But in the meantime, you can ask for it. And
you can ask for it, and I think some of these medical societies could probably say the available evidence, preponderance of evidence, suggests this is probably beneficial or may be beneficial, and there's no evidence it's harmful.
Yeah, what I'm trying to look at here, and I can't really find the data quickly that I want to see, is they have a statement in here that getting the vaccine early. So they have the statement that, those vaccinated before 15 years had the particularly 35% lower risk, those between 15 to 20 years, and then those vaccinated after age of 20, where they sort of talk about how that risk was, they don't have the same kind of improvement in their risk. I would love to go in more detail into that study and find out exactly how many women they had who were getting vaccination that late to know how well that was powered. And whether maybe if we looked at more women who were getting later vaccination, it might be possible to see that risk.
Some of that data are in this table here, table 3.
But the numbers aren't, are they?
Yeah, some of them have. Yeah, of 320 patients, 250 decided to be vaccinated. 7,904 women. Yeah, numbers.
But does it have the vaccinated
it doesn't have the
after age
breakdowns.
20? Yeah.
Well, I mean, it doesn't have everything.
Right, exactly.
Right.
And that's what Brianna is saying.
I'm trying to find the data on the people who are vaccinated after age 20.
Yeah,
you can.
That's just before the discussion, and it looks like it's in references 13 and 10.
Yeah, I'm looking at 10 right now.
Yeah.
But if you're listening and you have, if you are an OBGYN, get the ball rolling. Talk to your society.
Yeah.
Because I don't know how much attention this is going to get. It's a meta-analysis. It's in viruses, right? But it needs more attention.
Yeah.
Yeah. Maybe Daniel will mention it on his clinical update, and that'll catch a few more.
Good idea.
Yes. Well, Daniel often hears what we talk about.
Mm-hmm.
Like he just talked yesterday about the paper we did last week, mRNA vaccines make cancer treatment great again, which is an amazing paper in itself.
Yes.
Right? And he mentioned it last night. And so, many physicians are going to hear about that. So maybe he'll do the same thing, Daniel, right? You can do the right thing, Daniel? Okay. I know he listens at 1.5x.
That's exactly
terrifying.
the speed I choose, too.
But don't we sound like chipmunks?
Yeah, exactly.
No.
TWiV with the
when
chipmunks.
On the podcast app, when you do 1.5x, and YouTube will do this too, it brings the tone down correctly, and you could just, I watch like YouTube videos. Now, whenever you look up how to do something, it's never text. It's always, watch this YouTube video. Always watch those at 1.5x.
I did not know you could make the chipmunk go away.
Yes.
Yeah, speed it up. That's today's world. Yep.
Okay,
our paper today is from Cell. Human monoclonal antibodies against targeting A35 protect from death caused by mpox. I picked this for you, Rich.
Goody.
And Alan, the authors, please.
Yeah, so first author is Rihanna Fenton. Senior author is Camila Coelho. The group is kind of from everywhere. So Icahn School of Medicine at Mount Sinai, Scripps Research Institute La Jolla, NIAID at the NIH in Bethesda, Ghent University in Belgium, Yale School of Medicine in New Haven, University of Texas Southwestern Medical Center in Dallas, and Medical University of Vienna.
A couple of people on the author list who have been on TWiV. Bernie Moss, of course, Viviana Simon, Florian Kramer. Yes. Kathy, you have a summary?
Yes, it's pretty short. The gist of it is that we need to have better information about the monkeypox virus. We'd like to identify targets of antibodies, and so they're going to do this by using serum from people who had mpox. And they identified 3 high-affinity human monoclonal antibodies. They're named EV35 2, 6, and 7. So we'll be talking about 2, 6, and 7. And they target a particular protein in the monkeypox virus. And they block viral spread in vitro, and they protect mice against lethal monkeypox virus and vaccinia virus infection by both FC-dependent and FC-independent mechanisms, and we can get into that. And basically, they identify the viral A35 protein as a critical target, and they highlight that these antibodies against A35 would be promising candidates for treatments.
Excuse me, sorry. Rich, can you do a pox virus and explain, the extracellular envelope virion and the intracellular mature virion, please?
Sure. So first of all, pox viruses are large double-stranded DNA viruses with a complex virion that Vincent just asked about that we'll get into, not typical of the viruses that we ordinarily talk about. Replicates exclusively in the cytoplasm, which is one of the reasons, presumably, it's got a large genome because it has to encode a whole bunch of proteins that ordinarily a virus would cop from the nucleus, right? But it avoids the nuclear bureaucracy in the process. The most notorious, there are pox viruses of all sorts of animals, not only vertebrates, but invertebrates as well, that look similar. And there are multiple different pox viruses in individual species as well. The most notorious is smallpox, that devastated humanity for decades, if not millennia. Centuries, until the advent of the first vaccine, vaccinia virus, which was a related pox virus that didn't cause nasty pathology, called now vaccinia virus, that you could infect people with through their skin and cause a single lesion. By the way, typically these viruses cause a systemic infection, and then you blossom out in these lesions as long as a couple of weeks later, which is important. You feel sick, really sick, for a fairly long period of time, 10 days to 2 weeks, before the symptoms evolve that actually says, oh, you've got a poxvirus infection. And I think that's relevant. At any rate, vaccinia virus introduced through the skin gives you a single lesion and an immune response that's cross-reactive with smallpox. So it was an extraordinarily successful vaccine that ultimately resulted in eradicating smallpox from the planet. But there are other human pox viruses around, most notably, let me see, I can say monkeypox is the virus, right? Mpox is the disease. Mm-hmm. Misnamed. It's originally a rodent virus, but it's actually, the most recent versions have maintained a transmission chain in humans. We've had epidemics of this recently, but they're closely enough related to vaccinia and to smallpox so that a smallpox vaccination with vaccinia can effectively reduce or prevent mpox. All right. The particle is weird. It comes in 2 forms. The simple form is called intracellular mature virus. and it has a single bilayer proteolipid membrane on the outside. That's, well, I called it simple, but it's a single bilayer. It's not simple. It's got a whole bunch of virus proteins in it, and it is unusual as membranes go. Matter of fact, the protein has as much to do with anything. You can suck the lipid out of the particle and inactivate it, and then put the lipid back. The particle's still intact. You can put the lipid back in and recover infectivity. That's how weird this membrane is. Okay, the protein holds the whole thing together. That particle is infectious, and it is thought, and it's really tough. You can dry it out, which was important for the vaccination program because you could freeze dry the virus and still recover infectivity by rehydrating it. And the thinking is that that particle is the thing that provides for long-term transmission from host to host in nature. So you die loaded with virus, or you shed it on bed sheets or whatever, and that particle can tolerate a lot of environmental insults and still infect another individual. During the course of the virus infection, and I refer you to episode 68 of TWiV, Ode to a Plaque, my absolute favorite episode ever. During the course of infection, the first thing that's made inside the cells is this intracellular mature virus. But then that thing acquires a second envelope that also has virus proteins in it, about, at my last count, according to a review I looked up written by Jeff Smith some years ago, but I think is still valid. I think there were, this was from 2002. Holy cow, that was a while ago. 7 virus proteins in the extracellular envelope. And in fact, it acquires this as a double envelope inside the cells and then migrates to the undersurface of the membrane, and the outermost of those 2 additional membranes fuses with the cellular membrane, so that now you have sitting on the cell the IMV wrapped in another membrane. That's called the extracellular envelope. And that virus particle is thought to be it's actually perched on the end of actin filaments that can actually project this thing out and deposit the particle in a directed fashion on an uninfected cell. That's what episode 68 is about. And the proteins in that extracellular envelope particle are important for the infection in that case. They cause the binding and fusion and depositing into the cell the intracellular membrane virus. So the thinking is that that extracellular envelope virus, and this is really important for this, is what's responsible for spread from cell to cell during infection. So the extracellular envelope virus is responsible for spread during infection. The intracellular mature virus is responsible, this is oversimplifying, but intracellular mature virus is responsible for transmission from host to host. I think either particle can do both, all right? But those are their the thinking is those are their main roles.
I was just going to ask if they had different receptors, or if there were sort of differences in how the entry worked.
Yeah, that's I'm not as up to date on this as I should be, and it's really complicated because this is not this simple one receptor, one protein thing. Take it up. There's like a complex. There is an entry fusion complex on intracellular mature virus that has like 20 proteins in it, and there's binding to at least 4 different cellular proteins, and the actual process, I'm a little dated on this, but the actual process by which that entry fusion complex catalyzes the entry of IMV into the cells is not incompletely understood. And when it comes to EEV, I'm even less clear as to exactly how that happens. But yes, the viral proteins that are in the membrane are implicated in and probably important in the binding and uptake of the virus into the cells by different mechanisms for EEV and IMV because they're different proteins.
So antibodies are important for protection, and they know that from the smallpox vaccine, which induced antibodies that could persist for a long time. But in terms of mpox, the Jynneos vaccine isn't very good at inducing antibodies, so they say we need to do something better. And in this paper, they want to make therapeutic monoclonal antibodies, and they're focusing it on the A35 protein of the extracellular enveloped virus, which is, according to the authors, has recently emerged as a promising vaccine candidate. And this protein is a major target for neutralizing antibodies against orthopox viruses. And so what they do is they have a individual who has recovered from mpox with high IgG titers against the A35 protein. They identify 17 B cells, memory B cells that are specific for A35. They clone out the paired variable heavy and variable light B cell receptor sequences, and they use them to make 8 monoclonal antibodies against mpox A35 protein. They show that they bind, as do the antibodies from the original convalescent person. They also bind to A33, which, Rich, is the ortholog of A35 in
Correct.
vaccinia. Is that
Yep,
correct?
that's correct.
And they're all IgG1. And they look at the affinity, because they say, really good therapeutic antibodies bind from intermediate to high affinity to their targets. And they have EV35, 2, 6, and 7. They bind with high affinity to both mpox, MPXV A35, and vaccinia A33. Okay, so let's do some biology with these antibodies. In vitro neutralization capacity. They do a comet assay. Rich, what's a comet assay?
This is so cool. This is plaque assay on steroids, and I think it may be unique to pox viruses. Somebody out there may be able to correct me, but so pox viruses, you can do a plaque assay. Ordinarily, you do a plaque assay under a semi-solid medium. Okay, so to prevent virus from leaving one plaque and causing a secondary plaque, because you want to count the initial infections. But it turns out that pox viruses mostly stay very closely cell associated. So you can actually do a plaque assay under liquid and not bother with the agar overlay. But there is a certain amount of virus, EEV form, okay, that can leave that plaque and spread to elsewhere in the medium. And I don't know if it's something to do with, the situation in an incubator or whatever, but the secondary plaques sort of form a streak, and often in a kind of an arc out from the primary plaque. And it looks for all the world like a comet, where the plaque is the comet and the secondary plaques are a tail. So if you do a plaque assay under liquid, you can do that, and it is commonly used as an assessment of whether whatever therapy that you're using, be it a drug or an antibody in this case, inhibits spread. You do the same plaque assay where you would ordinarily see comets. You do it in the presence of your antibody or drug, and if the comets go away, that's an indication that whatever it is treatment you're doing is inhibiting spread. Really simple assay.
So you see, if you're inhibiting spread, the tail of the comet goes away, but you still have the body.
Yes, the tail of the comet goes away.
Yeah, and that's what they do here. You can see it. There's a very nice figure showing the comets.
You got to blow up that figure. I did that. I blew it way up, And yes, indeed, you can see the comets. They're really cool.
You can see comets with other virus infections under liquid. I've definitely seen them with adenovirus infections at low multiplicity.
All right.
Yeah, you said something about the incubator. It's interesting. If things are vibrating a lot, which happen in some labs, it will make the comets more eccentric.
Yeah, that's kind of what I'm thinking.
Right. Anyway, they do inhibition. They do a comet assay with these antibodies, and they reduce comet formation. So they're saying these antibodies reduce viral spread. I guess they don't eliminate plaques because the virus remains inside the cell, right?
Yeah, so I'm a little unclear on this, but certainly there is cell-to-cell fusion to some extent, in which case the virus just passes from one cell to another. And some pox viruses are better at that than others. So you can get, depending on the individual characteristics of the virus, you can get plaque formation without any EEV formation at all. They're usually smaller. Some of the plaque formation is EEV, some of it's fusion. I also don't know whether all of the EEV, in very close cell-to-cell contact, it could be that EEV could cause spread and yet be somehow shielded from whatever therapy you're doing. That's right. I'm stretching it there. I'm making that up.
So spread is almost like you're almost talking about sort of extracellular viral spread instead of intracellular viral spread.
Absolutely.
Yeah.
Yes.
Yeah, that's why the antibodies can affect it, right? Okay, so these seem to be neutralizing in a certain way, right? A specific assay. So then they look in mice. They see if you give antibodies to mice and then infect them, what happens? So they're using a vaccinia virus, which makes luciferase, so that they can take pictures of infected mice. So they infect they first give the mice intraperitoneally some of the monoclonal antibodies, 35-2-6 and -7. The control groups get a monoclonal against SARS-CoV-2. And then the next day, they challenge them intranasally with 100,000 PFU of the vaccinia virus. They measure weight loss and survival for 2 weeks. So the mice treated with A35, they have transient weight loss, and they gradually recover. The SARS-CoV-2 and the PBS controls, they keep losing weight until they die.
They all turn into tombstones by day 13.
That's right. They're all finished. And then they want to look at viral load in the mice and dissemination. They do bioluminescence imaging at different times post-infection, and you can see the inoculum and whether it reproduces at the inoculum site, the upper respiratory tract, and then you can see if it spreads elsewhere in the mice. And it's basically the virus inoculum is eliminated in all monoclonal treated mice between days 8 and 10, and viral spread, which happens in the control mice to other regions, doesn't happen in the monoclonal antibody treated groups. You can see that in the picture.
On the off chance that somebody might actually look at this paper, it is open access, and we're talking at the moment about Figure 3, and I have a couple comments about it. One is that in Part C is where they're showing their survival curve, and then on the right they start showing the survival percentage, and they do this for every one of their subsequent survival curves. And at first I couldn't even figure out what they were doing, and then really I don't know why they're doing it, because if you can read the y-axis on these curves, you get the same information that you get with these funny little,
the little
I don't even know
arcs.
what to call them, rainbow bars or something. They're strange. And the color discrimination between the different antibodies is not very good. I would love it if they would just use primary colors instead of shades of blue and dark brown,
they whipped
purple.
out the J. Crew catalog, and you've got, I don't know, plum and blueberry and I don't know what that would be, turmeric and
Yeah. And then another comment, just if you've not ever looked at these bioluminescence studies, vertically each mouse is assayed at a different time after infection. That's the beauty of these, is that you're not having to euthanize the mice at each day. You can look at them. And so you'll see the same mouse vertically and how the infection is progressing. And then my final comment, which is mostly for the authors, I hope at least one of the many authors is listening. The last part of figure 3, there is discordance between how the figure parts are labeled, their letters, H, I, J, K, and L, and the figure legend and the text. It's a mess. And I spent way too much time trying to figure out why figure H, as described in the text, was actually talking about protein flux, which was clearly something about the bioluminescence. And then finally I figured it out. So some kind of correction, maybe it's already out there, but some kind of correction of that. And it needs to be carefully looked at for the entire section about figure 3.
Yeah,
anyway.
Photon flux is the...
photon flux, yeah.
Yeah, it looks like what the things that are H and I in the figure are E and F in the figure legend.
Yeah, it's a mess. It's like they renamed the figure parts and didn't do it
I think there was an
consistently.
edit that didn't quite take in both places.
Wow. And
it's compounded by the fact that the figure legend is at the bottom of the following page, which is a cell layout issue rather than the author's. But
right.
All right. So far we have comet inhibition and protection of mice, but this is done with vaccinia. Now we're going to do it with monkeypox virus. So the same experiment. They treat mice with a monoclonal. One of these monoclonals from the paper were a control, and then a day later they intranasally infect them. The mice that get the control treatments, they have severe continuous weight loss for 7 days. The EV35 treated mice, they lose about 16% of their weight. The mice that get EV35, 6, and 7, they're 100% protected against infection. And one mouse in the 35-2 group died on day 11. So that's a 75% survival rate, but none of the control mice survived. So slightly different results with monkeypox virus than vaccinia. Okay, now we are giving the monoclonal before infection, right? But in the real world, if someone comes with monkeypox, with mpox, you can't turn the clock back before the infection and give them the monoclonal. So you have to do
and you can't really give
post-exposure.
monoclonals prophylactically because they won't be around by the time the person gets infected.
Yeah, unless you make a 6-month lasting monoclonal.
Right, but that's
we're not talking about that here. So they want to look at the therapeutic efficacy. Once you have disease, can the monoclonal help? So they use these 3 monoclonals again. They give 150 micrograms of each 24 hours after infection with 100,000 PFU. Again, this is back to vaccinia virus now. The animals that got the SARS-CoV-2 antibody, severe weight loss and death. And the mice treated with the A35 monoclonals, moderate weight loss. And survival rate was 87.5% for 35-2 and 7, and 75% for the other monoclonal. So they work therapeutically. It's 24 hours. I don't know how much longer you would get, but maybe the monoclonals can last a while, so maybe it would be a little bit longer. Okay. Then they want to know if there are any complement-mediated activities of these monoclonal antibodies, so complemented collection of blood proteins that can collaborate with antibodies to, in this case, prevent cell-to-cell spread of the pox viruses. So they did a plaque reduction assay in the presence of or absence of 5% guinea pig serum, which would provide the complement, right, which is in the serum. And they find that in the presence of complement, their monoclonals show a reduction in the number of plaques and a decrease in the size of the viral foci compared with the control wells. And if you have only monoclonals, you lose the comets, but the plaque size remains the same. So looks like complement is doing something.
It's like complement complements the antibody
Yes.
Yes.
as its name.
Time out,
Yep.
because I was thinking about you, Brianne, as I was reading this.
Okay.
So how does complement do this mechanistically? Do you know?
So there are a few different things complement can do. It is going to be activated when the antibody is bound to the virus. That may lead to things like opsonization, so that virus may be cleared, or it may agglutinate the virus. Theoretically, it could also help with neutralization, more steric hindrance of whatever is going on in terms of entry. So it could be a bunch of things.
But acting on the virus, not on the virus-infected cells?
It maybe could act on the virus-infected cell in terms of triggering it to be killed by an NK cell.
Okay.
So there's possibility of ADCC. So it could be any of those things. Yeah.
Handy stuff, complement.
It is.
So they decided to measure ADCC, antibody-dependent cell-mediated cytotoxicity. So an antibody is bound to the surface of a cell, possibly via a viral protein, and then other cells, like Brianne said, like NK cells, can come in and kill that cell. So this requires an interaction between the FC portion of the antibody and FC receptors. So they measure that, and they say, yeah, these antibodies bind the FC receptor, so they have the potential to participate in FC-mediated mechanisms like ADCC.
Right. And that could also help with the opsonization, since you'd need an FC receptor to do phagocytosis.
Right. So then they decide to introduce LALA mutations, amino acid changes into the FC region of these antibodies. We talked about these recently. You were here, Brianne.
Was it on immune?
Yeah, maybe it was
That might have
immune.
been immune.
Maybe it was immune. But I saw LALA coming up again, yeah. So that you can change the FC sequence of the antibody. It will no longer bind the FC receptor. So they introduced these changes. They confirmed that it blocks binding. And then they give these altered antibodies. They're not mutated antibodies, folks.
They're altered.
They're altered antibodies. Why can't people get this? There's so many authors on this paper. Oh, well. Altered antibodies were given to the mice, same conditions that we did before. They give the antibodies and they challenge. And this is, again, vaccinia virus. This, even with La La Land, the antibodies still fully protect the mice. They get some weight loss, but they live. So there is some effect of removing the FC functionality, but in fact, it differs according to the antibody.
According to which antibody?
Which antibody it is, but it's not the whole story here.
Yeah, so perhaps many of those mechanisms may be at play.
Yes.
That we've mentioned.
So this is why there are a lot of authors on this paper. They solve the structure of these antibodies bound to the A35 ectodomain. All three antibodies. This protein A35 is a homodimer, and at the dimer interface there's a groove, and that's where the dimer groove it's called, and that is where these antibodies bind. And they say this is unique because before, and the antibodies, each arm binds a different subunit of the dimer, right? And they said this is unusual because previous antibodies have just bound one of the two copies, and they don't bind in the groove either. So that's cool that this is interesting. And they can identify, of course, the parts of the antibody that interact with the antigen. They know the germline configuration of the antibody combining sites, so they could look at the residues that have changed by somatic hypermutation, right? And they could see that, hypermutation has led to additional interactions of the antibody with the antigen. We don't need to go into that so much, but that's very cool that you can do that. They do that for all these antibodies.
So I just have to make the point in this structure analysis part they talk about in one instance, each antibody binds with a large buried surface area, parentheses BSA. Okay.
Oh my gosh.
They did not need to abbreviate it because that's the only time that
it's
I can
used.
see that they used it. But, after us talking last week about abbreviations,
And they
And
chose an
BSA is
abbreviation that's widely used
Yes.
for something completely different.
It is widely
Yes.
used.
Yes.
Bovine serum albumin.
And then just now that I put my glasses on too, a comment that the fonts in this are really tiny, even in the graphical abstract. They could have been larger. And so that when I misread photon flux for protein flux, I could blame it on the fact that I didn't have my
That's a
glasses
cell
on.
layout issue.
And it's just really, really tiny font.
It's
they
cell
could
saying
make-
you can only
no,
have
it's
X
not
number
just
of
cell,
pages.
Alan.
Yeah.
People have to design their figures
Right.
with bigger fonts, and they can do it if they think about it or if the reviewers call for it.
But
Yeah,
it's also a product of the journal trying to cram things into fewer pages and saying, no, we can't give you a full page for this figure or that kind of thing. Yeah. There's
Well, in
blame
fact,
to go around here.
Also, graphing programs, the default font is rather small. I found years ago that I had to really bump it up to get it visible, right?
Yeah.
You can't just go with the default.
Yeah, and people around here at Michigan will tell you that I'm known for the fact that if I give comments on their seminar, it's almost every slide. The fonts need to be bigger. The fonts need to be bigger.
What a surprise,
They
Kathy.
usually
Yeah.
need to be bigger.
They usually
Yes.
do.
Yeah.
Okay, to the two last experiments, then they ask, are these epitopes recognized by these 3 monoclonals? Are they relevant in humans, and do they have anything to do with improved clinical outcomes? So they do competition ELISAs, binding assays of the antibodies to the antigen, and they have sera from people who have been vaccinated with DryVax, the first generation smallpox vaccines, then Jynneos, the current smallpox vaccine. They have sera from people with acute or convalescent mpox infection. And they find that the neutralization, so they can tell that the neutralization is targeting this A35 because they're doing competition with their antibodies. And it's that neutralization is much higher in people who've been infected compared to the vaccinated people. And, they think these antibodies these epitopes are important. So that's not good. It means that and we know that DryVax doesn't work so well, right, against mpox. And so that's why they think that we have to work on this. And the last experiment is they these are slightly different epitopes in the 3, and they look at these convalescent sera and vaccine sera, and they show that combinations of the antibodies are correlating with improved clinical performance.
So people who naturally develop these antibodies when they're infected correlate with better outcomes.
So there you have some interesting monoclonals. They say these could be useful for, severe cases, especially immunocompromised individuals who are very difficult to treat. And they point out that the drug tecovirimat and brincidofovir have not really worked that well in clinical trials. So monoclonals could be very useful in future treatment. And they say because this the FC, they still work without the FC binding. Maybe we could make single chain antibodies or other formats that are independent of FC, which are much easier to manufacture and they're cheaper.
Nanobodies.
Nanobodies, right?
So I have a comment about this statement that although promising in vitro, current drug therapies such as tecovirimat and brincidofovir have proven insufficient clinical trials. We touched on this a few episodes ago, I think, when the tecovirimat clinical trial came out. And I want to remark, first of all, the tecovirimat has a similar mechanism in that it inhibits spread. In fact, it interacts with another one of the EEV proteins, which in vaccinia is called F13, and inhibits the actual wrapping of the IMV particle in the double membrane that ultimately becomes EEV. So it inhibits the formation of the extracellular envelope and therefore inhibits spread. And when people first started working on this and thought, okay, we're working with a drug that's going to inhibit spread, the thought was, man, that's never going to work, okay? Because you really got to inhibit the replication, not the spread. But it turned out that in all of the in vitro models and all of the animal models, tecovirimat was a miracle. And when this trial came out that said that in the field, in treatment against monkeypox infections, it relative to controls, it was not effective. I was really surprised because all of the laboratory and preclinical data were so good. But going back to my initial description, by the time you show up at the clinic and you've got lesions that look like that say you've got monkeypox, you could be a week or two down the road and you've already got a systemic infection. And at that point, an inhibitor of spread, and maybe even any inhibitor of all, is not going to do you a whole hell of a lot of good, okay? As with many of these antivirals, timing is absolutely critical. You got to deliver it the minute you see a problem. And I think this work is great, but it's going to that's going to be a factor in using this kind of stuff.
Yeah, I had two things. Did we mention that this A35 groove epitope is highly conserved?
Yeah.
Yeah, weird, huh?
Yeah, I mean, so this, whatever we get out of this, could be kind of a pan-pox type of approach that, you could stop a bunch of things with. But yeah, obviously Rich's point of you may not be able to treat mpox with monoclonals because by the time you diagnose mpox, it's too late to stop spread. But they discuss a little bit, what this might mean for vaccine design, and they suggest designing vaccines that would target, I guess, the B-cell germline epitope, and then that, because hypermutation is so important. So I thought that was kind of an interesting approach. I don't know if that's generally been done either deliberately or just that's the way the vaccine ends up working.
Yeah, I think that's a goal in a number of different vaccine trials or vaccine designs at this point.
I would strongly recommend that they not be called germline targeting vaccines. Mm-hmm. That's the term they use here, and I just...
Yeah.
I have another...
Rich, it's possible that these monoclonals won't work in people, right?
Yeah.
Because of what you just said about tecovirimat.
I have another observation about vaccination against pox viruses in general. I think immunity to pox virus infections is an extraordinarily complex issue, in particular because the pox viruses have so many immune countermeasures and et cetera. So we've already made the point that some of the vaccines, are, not as good as you would like them. I should point out that the problem is that the best vaccines, like vaccinia against smallpox, have side effects because you're infecting somebody with a live virus, and at very least, in that case, you get this nasty blistering lesion that nowadays nobody wants to deal with that kind of stuff. Not only that, but the lesion is full of virus that can spread to somebody else, all right? Yeah, in a situation where the whole planet is infected with this thing, you got a 30% mortality rate, that sort of side effect is not an issue. But in a low incidence situation, that kind of side effect is undesirable. It's a risk-benefit ratio thing. So you look for better vaccines like this. Jynneos is a highly compromised virus that's been passaged. It's a derivative of this MVA thing. It's been passaged in culture forever and is missing a whole bunch of these immunomodulatory genes and a bunch of others. And it turns out that although it's really safe, it's not as good as a vaccine. I should say people are also working on messenger RNA vaccines, but a single antigen is probably not going to cut it. The frontline mRNA vaccines, as I know, the best candidates, have like 4 different genes in them, 2 from the EEV, I think, and 2 from the IMV membrane. A35 or A33, depending, is one of these. Okay. But I don't know how well those are going to work out. I want to point out that I'm probably breaking some sort of law here, but I'm going to plug a company called Tonix Pharmaceuticals that, if you'll recall, this must be years ago now, a paper came out from Dave Evans' lab where they recreated horsepox
Yes.
from synthetic DNA fragments. The reason that they did that was because, and we ought to go back and do this history again, the thinking is that what we know now as vaccinia is most closely related to something that we have called horsepox. Now, I put that in my mind in quotes because there's only ever been one isolate of what we call horsepox from a horse in Mongolia in the 70s or something like that. So I don't know whether that virus is something that used to routinely infect horses, because it doesn't anymore, or whether it was just some random pox virus that this horse picked up. Nevertheless, that virus looks to be the foundation of what we have called vaccinia. And these guys at Tonix thought, maybe we ought to go back to square one and use the virus that was most effective initially. We'll do horsepox. They couldn't get horsepox from anybody. So what do? You call Dave Evans, who knows how to do this, and say, will you make this for us? And they say, sure. So now there's this company called Tonix that has done a bunch of preclinical studies with that virus that is a reconstructed horsepox. And in many ways, that looks to be the best vaccine, could be potentially the best vaccine out there. I don't know the details, so I don't know the safety factors involved. For all I know, it's going to be one of these things that's got to be administered by scarification, gives you some lesions. I don't know how bad they are, but this is a tough nut to crack.
But it's not a vaccine, it's an equine.
What are they testing it against? Mpox, I presume?
I do not know. I'd have, you can look up Tonix Pharmaceuticals,
All right, we'll look it up.
and they'll tell you the whole story.
Okay. If they've published anything, maybe we should look at it.
Yeah.
Okay. Let's do some picks of the week. Brianne, what do you have for us?
I have an article from New Scientist that I really enjoyed reading. I clicked on as soon as I saw the title, called Civet Coffee, The Real Chemistry Behind This Bizarre Luxury Drink. Some people may be aware of the palm civet. I talk about the palm civet in some of my emerging infectious disease classes when talking about SARS-CoV-1. And every time I always end up somehow on a sidetrack talking to my students about civet coffee, which is one of the reasons why palm civets are trafficked. And they never believe me that it's a real thing. And so this article...
Oh, come on. Dave Barry wrote about this years ago. It's got to be real.
Yeah.
Yeah, but current college students don't.
I know.
Don't know big Dave Barry.
Yeah, so this is a New Scientist article. It's reporting on a scientific reports paper that actually looked at coffee beans that had been passed through a gut of a palm civet versus other coffee beans and did chemical analysis to see what was different. And they found quite a few differences that they thought could explain some of the differences in taste in this luxury coffee. I've never consumed civet coffee. I never really thought about it, although when I was reading this paper, I was like, oh, it's talking about, reduced bitterness and reduced acidity. Maybe that would be a good thing.
Yeah, $1,000 a kilogram. Just pick up a bag.
So I did think that was sort of interesting, but I was very excited to use this article in the future to prove to my students that civet coffee is a thing. And in fact, people did a scientific study on what makes it taste perhaps different.
So, how do they get the civets to eat these without chewing them?
I don't know enough about that, but I think that they might be partially digested and maybe even partially chewed.
Oh, you don't need a coffee grinder.
So coffee beans are. I'm not an expert on this, but last time we were in Hawaii, we toured a coffee farm. Okay, because they're all over the place, and coffee beans are one of these things where the bean itself is the seed inside a really pulpy dude.
Yeah.
Okay. So I'm guessing here, but my guess is that the civets are after the pulp and swallow the seeds, essentially.
Maybe.
That
Maybe.
could very well be the case. Yeah.
It's like a cocoa bean.
Yeah.
Mm-hmm.
So cocoa beans are actually fermented
Yes.
on farms, and the boxes that they're fermented in and the burlap covers have the microbes that contribute to the fermentation. It's kind of a random process, right?
Yeah, and that's what they talk about in this article. They're like, maybe if we could figure out what this fermentation is that's giving these flavors, maybe we could do it in a lab and not have to worry about working with civets and also make it cheaper. Yesterday on TWiV, we did a paper where they did that. They identified the microbiome of good chocolate, high quality chocolate. They figured out, they identified 9 microbes, I think 4 bacteria and 5 fungi. They put them together, and according to their tasters, it was great chocolate. I'm finding that paper before my micro
Can you
class
imagine
next semester. That's awesome.
I mean, the value of that, and plus you could probably manipulate it and make any tasting chocolate that you want. Okay, now the other paper we did, just very quickly. It turns out in Bulgaria, for thousands of years, they've made yogurt by adding 4 fire red ants to milk.
Whoa.
In a bottle. They put the bottle in the ant colony to keep it warm. It ferments and makes beautiful yogurt. And what they did in the paper, they identified the microbes in the ants and the acids, the formic acid and the acetic acid, and proteases from the ants that all contribute to making yogurt. Cool. This is incredible.
Definitely looking for that episode.
Okay, sorry. Kathy, what do you have for us?
I picked something that we haven't done for a while, and that's just an Astronomy Photographer of the Year awards. And when you click on it, it seems like there's a fair amount of ads and things that you go through, but they're just some really nice photographs of astronomical kinds of things. And in fact, the second one is a comet.
Hey, look at that.
Today's comet plaques and things like that. So just enjoy looking at astronomy pictures, because those of you who have Astronomy Picture of the Day as your browser homepage know that it hasn't been refreshed for the last month because it's a government contribution to the pages. So anyway, so now you can look at these instead.
The overall winner, what is that sitting on? It almost looks like a Buddha statue sitting on the rock, or is that just more rock?
I think it's
probably
just
more rock.
Yeah.
I
wonder about that too. Yeah.
Wow, it's really nice.
Well, Dixon showed up just for this.
Right.
To see the photography.
Yeah.
Cool. Rich, what do you have for us?
So I decided to basically make this a memorial to my sister-in-law, Kathy Swan, who died of a cervical carcinoma at the age of 34.
Oh my gosh.
In 1992. And when I did lectures on papillomaviruses in medical school, I always used to start with a clinical case. And I used Kath very effectively as my clinical case for the papillomavirus lecture. She was a rheumatologist who had just completed her medical and residency training and was working in the at the Beth Israel Hospital in rheumatology during her disease and ultimate death. And she had a reputation for being an extraordinarily compassionate physician. And to honor her, after her death, they created a program called the Catherine Swan Ginsburg Humanism in Medicine Program. By the way, for those docs out there, Swan is the swan as in Jeremy Swan. That's the swan in the Swan-Ganz catheter. So she is the daughter of Jeremy Swan as in Swan-Ganz catheter. I'm his son-in-law for all that matters. At any rate, she was such a compassionate individual that they created a program called the Catherine Swan Ginsburg Humanism in Medicine Program to promote her sort of compassion and humanism in their medical residents and their students, honor those who keep up with that tradition, and have actually lectures and seminars and speakers who talk about maintaining compassion and humanism in medicine. So I thought I would just ping that and actually ask people to honor that compassion and humanism in medicine. This is a big deal. It's an important discipline at the University of Florida. They used to they had classes on paying attention to the patients, not just the disease. So there you go.
Not all physicians are created equal.
Not all
in that
physicians are
aspect,
created equal.
Yep.
And Rich, it was her paper earlier in the show notes holding the lobster.
Yeah. So that was that picture was taken. Must have been in Sun River. Remember Bend?
Mm-hmm.
Okay.
Mm-hmm.
Sun River at the family vacation house. Must have been the Christmas of 1991. About that would have been 10 months before she died. And she had just, she'd been, this is the way this disease goes. She'd been diagnosed with a cervical dysplasia 10 years earlier. That was treated with a procedure that wound up not getting it all. And 10 years down the road, she wound up with a cervical carcinoma. She went through all sorts of horrendous treatment and looked to be clean. And we're going to come together for Christmas and celebrate all together and that kind of stuff. And right before she came out for Christmas, she had a scan that revealed that she still had some cancer left. And what's remarkable about that picture is the smile on her face.
Yeah.
If she had a vaccine back then,
Yeah.
that the procedure would have saved her life.
That's right.
Quite likely.
That's right. When she originally was diagnosed, that would have been 1980. And at that point, people were still questioning whether papillomaviruses actually caused cancer.
Right.
And we knew nothing about the different types. So during the course of 20 years giving these lectures, I was able to see from not even being sure what the agent was to developing a vaccine that'll prevent the cancer. Unbelievable.
What I find particularly egregious is that there are companies suing Merck, the manufacturer of Gardasil 9, claiming that it causes damages. It does not.
It does not.
Saves your life.
Yeah.
Alan, what do you have for us?
Well, this is a Halloween, kind of a Halloween-themed pick. It was recent, and I thought, I'm going to pick this for the Halloween TWiV. So I'm linking to a science article, news article about this that has a video right at the top. You can watch the video, which is creepy enough. But if you follow the link to the paper is open access, and the paper is really interesting. So there are these little roundworms that are parasitic on flies. And if you're a roundworm and you live in the soil, how do you get to a fly? Well, you could wait for it to land on you and, grab onto it. But these worms have the ability to jump. So they're waiting there in the soil, and they, I guess under certain circumstances, maybe occlusion of the light or something, they will jump. And they jump many, many times their body length into the air. The problem is, if they don't successfully hit the fly, they're going to fall back on top of the soil and probably die of dehydration. So this is a really high-risk maneuver for the worm. How do they manage? But they're a successful parasite. They manage to get to the flies a lot of the time. And so these researchers hypothesize that maybe static electricity is helping. Because flies, like airplanes and helicopters, they're flying through the air and they generate a positive static electricity charge. So as they fly above the ground, and this has been shown with bees going, and one of the reasons they pick up so much pollen is because they're positively charged, the flower is grounded, and the pollen, sticks to the bee, just like your hair on a balloon. So they tethered a Drosophila fly and gave it to a copper wire and gave it various degrees of charge. And then they had the worm on a filter underneath it allowed to jump. And they discovered that, in fact, when the fly has about the level of charge you'd expect when it's flying, if the worm jumps up and is anywhere in the ballpark of the fly, it goes zip and gets extracted right up to the fly and sticks.
Wow.
And starts its parasitic infection.
It's cool.
Yes.
Amazing.
Nice.
I love it.
So worms jumping out to infect the flies.
Awesome.
My pick is a nature feature, news feature called How to Get the Best Night's Sleep, What the Science Says. I'm always looking for ways to get a better night's sleep. Of course, the best way would be if I could turn back my clock 50 years. Then I would sleep great, but I can't. And so this article gives you some clues about that, which some of them have come from studies of circadian science, right? Studies of our biological clocks. But they say 3 basics: contrasting light and dark, consolidating meal times, and keeping sleep times consistent. Simply taking a walk outside during the day and reducing light exposure in the evening could have a great effect, because light is a big trigger for the circadian systems. And also, calorie intake is an important part. We're not actually equipped to be eating at all hours, apparently, although a lot of people do. And they say here, here's the adage: eat like a king in the morning, a prince at noon, and a peasant at dinner. The last meal should fall 3 hours before bedtime, with no subsequent drinks, snacks, or caloric drinks. And of course, the food matters. Stay away from high sugar food, saturated fat.
So if you're going to have a drink, you should have it at breakfast.
And alcohol is totally disruptive. Yes. So they say alcohol will make you fall asleep, but your sleep quality is quite poor.
And I'll tell you, too much alcohol not only makes you go to sleep, but then it makes you wake up.
Oh, yeah.
Mm-hmm.
And then they say eating plant-based foods during the day could improve your sleep. Anyway, a lot of good clues here. If you have trouble sleeping,
All right.
check it out. We have a listener pick from Braden. Hi, TWiV team. We've had a 30-plus C day crowned with a little sprinkle of rain here in Herrera. After hearing that Alan had let his pilot's license lapse for environmental reasons, I was inspired to share an organization called Elevated Access with you. They're a group of volunteers who fly people from states in which abortion and gender-affirming care are unavailable to states where they can get the care they need. Here's their Instagram page. I agree that general aviation is environmentally unfriendly, and sometimes having a pilot's license and access to a plane is the difference between a good quality of life and getting legally restricted care. Dan Savage, sex and relationship advice columnist and podcaster, recommends that people keep a viable supply of abortion pills in their homes. Even if, like Savage and his husband, they're never going to need the medication themselves, but can provide them, no questions asked, to loved ones who need them. He had a doctor on the show to defend Tylenol when that study hit the news. Maybe Elevated Access will expand to include obtaining vaccines in the not-too-distant future. I hope Americans won't need to fly to blue states to vaccinate themselves and their kids, access Paxlovid, etc., but it's always good to keep these options available. With the axe of hope in my hand, that's Braden, he says, I'm referencing Rebecca Solnit.
Nice.
It's really good.
Yeah, just to be clear, I'm not opposed to the existence of aviation or general aviation. And there are many, many people who have totally legitimate reasons to do it. I mean, if you're a flight instructor, if you're learning to fly to make a career of it, absolutely, go fly. My problem was that I didn't have a good excuse. It was, I was just doing it for my own entertainment. And I thought about becoming a flight instructor and kind of made some of the progress toward it, and then realized I don't actually want to teach people how to fly. But so, I didn't have a good enough reason. But something like this, or people who live in Alaska and, this is the way they need to go to get a medical checkup because the state's too freaking big. That's fine. That's absolutely fine.
Yeah.
You're missing the point here, Alan. Braden's giving you an excuse to fly it.
An excuse to fly. Yeah.
Yeah. All right. That's TWiV 1267 Halloween edition 2025. Did we ever have a TWiV on Halloween before?
Probably.
Must have.
I guess we must have, but I don't remember. We certainly didn't
I don't
have
know.
guests.
How frequently does any given day of the year fall on a Friday? Is that 1 out of 7?
Ask Siri or
No.
whatever.
It's something about also could be because of leap years, 1 out of 14. But yeah.
Yeah, I guess we've been doing it long enough that it probably happened before. Anyway,
AI says approximately once every 7 years, but it can vary.
so we could have had a couple.
Could have
Anyway,
had a
TWiV 1267. You can find the show notes at microbe.tv/TWiV. TWiV, if you have questions or comments or want to send us a pic, we love to hear pics from you guys. It's great. TWiV at microbe.tv. And if you enjoy these programs, we'd love your support. Microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. And
Thanks. It was fun being here.
Brianne Barker is at Drew University, Bio-Prof Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot, and may the force be with you.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com and turbidplaque.com. No comments, right, Alan?
No comments. No.
Thank you, Alan.
But thanks. So it's always a pleasure.
All right, it's a little oblique, I know, but turbid plaque,
Yes, turbid plaque.
You got turbid plaques, but no comments. I'm Vincent Racaniello. You can find me at microbe.tv or on Blue Sky, MicrobeTV with no dot. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.